Okay, I'd like to welcome my colleague Tom North. So I think most of you know Tom. Tom's the curator of the National Seed Bank at the Australian National Botanic Gardens, Canberra. He's got a 25-year history of working in seed banks for conservation and research of Australian native flora. His co-authors, his acknowledging hero Katie Guzzi and Leonie Coleman. Tom's talk is about saving a pea, practical conservation horticultural techniques lead to a successful translocation of the endangered small purple pea, Swainsona erecta. I have a particular interest in this little pea, being the one of the park carers that look after its source population on Mount Taylor Nature Reserve, and it persists in this very fragmented urban remnant despite herbivore grazing and the fires of 2003. So over to you, Tom.
Thanks, Kathy. And I'd like to acknowledge the Wiradjuri people on whose country this work took place. This is a bit of a walk through practical steps in saving this pea, the Swainsona erecta, and in particular the steps that include seed banking and horticultural techniques. So just as a bit of a backstory, the small purple pea once covered country on the western slopes of the Great Divide from Dubbo down through south down through to Ballarat. The Victorian populations are no longer are extant, and a lot of the other southern New South Wales populations occur only on railway easements, and most of those populations are declining due to high sea predation and low vigour in those populations. They're quite small populations. In 2019, the local land services from Central West and Central Tablelands received a grant from federal government and New South Wales governments to do work on conserving Swainsona erecta in their areas. They engaged an ecological survey team to model and then to do surveys across that area, and they were successful in identifying new populations across between Mudgee and Dubbo of quite significant numbers. So in 2019, those local land services also approached the Australian National Botanic Gardens to, with our experience, to undertake the translocation work on the small purple pea in those areas. And this is a bit of a promotional step to promote the work of ANBG and the translocation guidelines that came out in 2019. We borrowed heavily from those guidelines to produce a translocation plan for the small purple pea, covering a 4-year project. One of the first steps we did is just ensuring that we had all the licences lined up. So talking very early on with threatened species officers that have responsibility for that particular species, and also land managers, and making sure that we had those scientific licences in place for collecting, but then also for the translocation, and approaching land managers to establish contracts and perpetuity of contracts for managing the sites that we were identifying for translocation. The next step in the small purple pea conservation was just running through some of the steps that we need to do. So assessing the remnant populations, ensuring comprehensive sampling from across those populations to collect enough genetic representation, looking back at the literature on what had been done before and relying heavily on work that had been done by Andrew Young and Melinda Pickup on the genetic and cytology of small purple pea. Establishing a banking and conservation research, and then putting in place germination trials and understanding the biology, propagation for translocation with the ANBG nursery, and then augmenting those remnant populations. So one of the first things we did was assessing those populations and then trying to identify some of the threats to those populations and how we could remit them. So looking at declining populations, which populations were quite stable, identifying those for more significant collections, which populations were low in vigour, and we're understanding that through looking at recruitment and understanding the seed biology, and then which populations were subject to changing environments. So I'll just note that this project was quite a lot more straightforward in terms of the challenges that we were facing compared to the example that Alicia gave before. But we still ran into that, as a 4-year project, we still ran into some hurdles that we encountered, especially the drought started in 2016, but by the start of this project it was full grips especially across that central New South Wales, affecting plants through 2019 through to early 2020. Later collections were impacted by COVID-19 restrictions. We were in Canberra, the plants were up in areas from Mudgee to Dubbo, and so getting to those sites to collect proved quite difficult, and working around the timing of collections was significantly impacted by those restrictions around movement with COVID-19. The distance, being there on site, relying on local knowledge and having volunteers involved in the project became quite critical. And then, as I mentioned, with that risk assessment, the declining populations and the low vigour in those populations as well that we were collecting from. So in terms of collection strategy, again, to plug ANBG and their publications, the Flora Bank and the Plant Germplasm Conservation in Australia guidelines, guided how we set up our collection program for the pea. Again, mentioning that we aim to collect as much genetic diversity as possible from across all the populations and capture them in our propagation. And then actually being on time, bagging stems prior to fruiting, just after the end of flowering, to capture optimum time of collection and seed maturity. Once we'd done that, as you can see, that first year of collecting that we had in 2019, there was absolutely no seed to collect because of the drought. 2020-21 collections, plants had started to recover, and we had quite reasonable collections of about 8,000 seeds from across 21 sites. And that improved in 21-22, again, a very good year, and 13,000 seeds. Again, just collecting 10 to 15% of available seed from each site and population. The 2022-23 collection year was just a backup year for us, ensuring that from those sites that we hadn't managed to collect enough seed from, or we needed better representation, we went back to those to collect. And in all, we collected around 22,500 seeds over those 4 years. That size of collection enabled us to do quite a lot of work in understanding germination. One of the trials that we did, Lydia Guya and Gemma Hoyle undertook for me, was just basically torturing seed to get an idea of longevity and persistence, both in seed banks, but also ex situ, but also in soil seed banks. So this study, in comparison to a lot of the other grassland species, was showing that Swainsona has quite reasonable persistence, but then it does lose viability quite dramatically over time. The next step that we undertook with that germination work was the propagation, and I have a big debt of gratitude to Pete Pradell and the team at the ANBG nursery. Swainsona proved quite difficult in propagation. We weren't seeing a lot of vigour, and the germination was quite low. So even though we had about 22,500 seeds in the bank, we ended up using just more than a third of that collection in trying to get enough plants into propagation to supply the translocation of about 1,100 plants. But as you can see in those images below, what they did manage to do was produce healthy and vigorous plants once we got them going. And they were able to play around a bit with the soil media, and the image on the right that you can see there is one of the trials that Pete's been doing with long tubes, and Swainsona's proved to enjoy that, really, the ability to put down its roots. It's producing up to about 1 metre of root, which gives us a better understanding of what it's doing in the wild as well and how it persists. And I'll explain a bit more of that in a sec. So with the translocation, the first translocation occurred in April 2022 over 3 sites in the Central Tablelands and Central West, and we managed to plant 700 plants over that time. And one of the things that we realised, it was quite a good year, but with that came weed competition. So managing that competition became pretty critical to survival of the plants. So mulching with sterile sand or gravel around the plants proved highly successful, but also making sure that we dug down deep enough to enable that long taproot of the Swainsona to get down and do its thing. The second translocation occurred in April 24, and we put the remaining 500 to 600 plants in the ground over 4 sites on both private and public land. Over that time, over the 4 years of the project, a lot of the work was done by volunteers, and we wouldn't have been able to achieve as much as we did without them. Just to note, most of those volunteers came from our team at the National Seed Bank, our seedy volunteers and curation volunteers. And all in all, we had 44 volunteers involved at that time, providing 289 hours of work in the curation processes that we undertook. And then for the translocations, again, we had about the same 44 volunteers, but 136 hours during the August translocation and 92 hours all up in the April translocation. I've been relying on the local land services to do the monitoring, and so this is still in early stages. They've done 1 monitoring season in October 2023, and I was hoping to have the next tranche of data for October 24, but they haven't quite taken that yet. But doing the monitoring during flowering season makes sense, so we've aimed to do that every October for the next 10 years. First, a lot of monitoring has shown that we've got about 64% at least survival rate. The plants are actually growing and flowering, but with Swainsona it's not easy to tell whether we've got dormant or dead plants. So the plant does do this cycling thing of going into dormancy. Our next steps to do is to continue that monitoring of the translocation plantings over from the 22 and 24 plantings, implement the recommendations that we've learnt through the seed banking and horticultural steps in the actual planting, and apply them to, we're now involved in another translocation of Swainsona in the ACT. We'd like to investigate further and implement and monitor the management practices that are aimed at ensuring the survival of Swainsona and encourage recruitment and understand that better, but also undertake a seed burial trial in both the Central West and the ACT to better understand that seed longevity and persistence in the soil seed bank. So what I'd say in terms of lessons learnt, I'd say over a project, maintain your sanity. I do have a ream of correspondence between myself and threatened species officers to try and get our translocation plan right, and that ream goes back 4 years, thousands of emails to get that right, but I think we did get it right in the end, so persistence paid off. Having that comprehensive translocation plan was really important. Following up and having a post-translocation plan with site managers so that those sites are managed long-term for the survival of the species, and hopefully managed in a way that we do see recruitment in the future. What we found is that there is a lot of changes in government authorities around the country in personnel, so managing that and keeping communication going was very vital. And yeah, keeping that communication going between licensing authorities is also very key for the partners and everyone involved. So thank you to everyone that was involved. A special thanks to Pete Pradell, Micah Stevenson at the ANBG Nursery, Lydia Guya and Gemma Hoyle for their work on the seed longevity, and Mitch Corder and the ANBG volunteers that helped with collection, curation and planting, Katie Guys, Libby McIntyre and Stephen Pereira at Central West, and Leonie Coleman and Evelyn Nicholson at Central Tablelands. Thank you.
Thanks, Tom.